what are cell lines?
Good afternoon, ma'am.
Yeah,
Good afternoon.
good afternoon. So today we will discuss about cell lines as well as tumor cells. Okay, so first we will discuss about how to maintain and propagate cell lines and what are the screening techniques that we can do on these cell lines. So what is a cell line first? So cell line is a permanently established cell culture that will proliferate indefinitely given appropriate fresh medium and space. That means it is like there is no further differentiation or nothing like that. So it is established. And it, like tumor cells, they will proliferate indefinitely for longer periods of time, like HeLa cells. So they are cell lines, okay? Unless you don't give any media or required like appropriate conditions, they won't die. Okay, so if you give appropriate fresh medium and space, they can divide for n number of times. And a cell culture developed from a single cell and therefore containing of cells with a uniform genetic makeup. So all these cell line means it is starting from a single cell. Okay, so all of them, whatever the cells which are there in that cell line, they all will have same genetic makeup. There are no mutations or different genetic composition. And there is presence of several cell linkage, either similar or distinct. So we can between cell lines also, we can see some similar type of cell lines and some may be distinct. And stem cells are usually used in this cell line culturing. So after the subculture, the primary culture becomes a cell line and may be propagated and subcultured several times. So I told you, like till the first subculturing, that means from receiving from the explant, like a source tissue, till first subculturing, the cells are called as primary cell cultures. So once we do subculturing, they become cell line and they can be propagated and can be subcultured any number of times. And some species, particularly rodents like mice, then rats, they give rise to lines relatively easily, whereas other species do not. So usually if you see the cell lines also, they are mostly from human or mice, we can see, right? Rather than any other animals. So that means they are easily propagated. Okay. And no cell lines have been produced from avian tissues, and the establishment of cell lines from human tissue is difficult. So though we do it, like we do experiments on human cell lines, but to establish them, it is difficult. But why we have to do on human? Because it is the finally we have to treat or we have to study the mechanisms in human beings. So ultimately, though we perform the preliminary experiments in some animal models, but finally we have to do it in the humans. Okay, then only we can actually get the product or mechanism out of it. So there are different types of tissue cultures. They are primary and continuous. So primary tissue cultures are nothing but they are the normal cells cultured without any change in their division rate. So they are mainly from, I told you, like explant, directly from explant. So there is no, like they slowly they will divide and they are normal cells. In continuous cell lines or continuous tissue culture, we have finite and indefinite. Finite means a single cell type roughly 30 times of division enhanced by growth factors. That means we are enhancing its division rate by you applying some growth factors. They are like treated. Okay. Then indefinite. Indefinite means it is it is nearly the same as the finite. Okay, but the cells can divide indefinitely by transformation into tumor cells. Then they are called as cell lines. So here in finite, you see it is 30 times we are telling roughly. So that means after some maybe 30 plus or minus some number of cycles, but that will die in some or the other cycle. But in indefinite or cell lines, there will be continuous division. They are transformed cells, okay, like most of the tumor cells. So which will be continuously dividing for any number of years if the conditions are maintained properly. So cell lines are three types: normal, transformed, stem cell. So normal are nothing but they are taken from the tumor tissue and cultured as a single cell type. So directly taken from the tumor tissue and they are cultured. Then transformed. That means the normal cells, it is not the tumor cell, it is the normal cell which underwent some genetic change to form tumor. Okay. Then stem cells. So stem cells, yesterday we have discussed the whole class about stem cell. So stem cells are the master cells that generate different types of other cell types. So what are primary cultures? So cells when surgically or enzymatically removed from an organism and placed in a suitable culture environment, so that will attach and grow, then they are called as primary cultures. They have finite lifespan. They cannot be indefinite. And they contain a very heterogeneous population of cells. So you cannot just isolate. When you take a tissue and apply some enzymes or some mechanically, you are making them small pieces and you are separating the cells, you cannot imagine to get all are of same type. So because it's a tissue, you can expect different types of cells. So that is heterogeneous population of cells can be expected. And subculturing of primary cells leads to the generation of cell lines. So whenever subsequently we subculture them, then they will be transformed at certain point of time as cell lines. So cell lines have limited lifespan here and they pass it several times before they become senescent or they will die. And cells such as macrophages and neurons, they do not divide in vitro. Why? Because they are in G0 phase, neurons. Okay, so they cannot go back to G1 phase. They enter G0 phase, which is inactive. So they can be used as primary cultures. Then continuous cell lines. So most cell lines grow from a limited number of generations.
Excuse me, madam.
Yeah.
And macrophages?
So even macrophages, they will be like two types, monocytes and macrophages. So they will be circulating. So I will just check one, why they don't divide. Okay. I am sure about neurons that they enter into G0 phase. That's why they won't divide. But I will check with the macrophages and I'll let you know in the next class. Okay.
Okay, ma'am.
So in the continuous cell lines, most cell lines grow for a limited number of generations. Okay, because either they may get some mutations or they lose their ability to divide. Okay, and the cell lines which either occur spontaneously or induced virally are chemically transformed into continuous cell lines. So what any cell line which is normal, if we are inducing it with some virus or due to accumulation of some mutations, okay, they will get transformed into continuous cell lines like cancer cell lines and they will be dividing. So what are the characteristics of continuous cell lines? So usually if you see, we don't immediately after looking under microscope, we can actually differentiate what is a dead cell, what is a live cell, which type of cell, like that, if you are experienced. Okay, so there are some characteristics because we have studied general histology, right? So in that we have seen that each cell type has a different morphology. So like epithelial tissue, in that we have a squamous type, cuboidal type, like that. That means they will have some shape. And when we look under microscope, immediately on the first day of adding the cells, all cells will be round and they will be floating in the medium. But after 24 hours, all the cells will come down, they will attach to the substrate, okay, and they will come to its original morphology. And after 24 hours, if you see, you can easily notice that what type of cell it is, and you don't find any floating cells. But after some days, if you are not changing the media, or if there is any contamination or anything like that, and even because of overgrowth also, the cells will start floating. So the floating cells are dead cells. After 24 hours, before attachment, you cannot say like they are dead cells because they need time to get attached. But once they got attached, still if they are floating after some time, then they are dead cells. And through morphology also you can tell. So here continuous cell lines also will have some characteristics by which you can just tell whether they are continuous or not. They are smaller, mostly rounded in shape, less adherent, that means they will be floating, with higher nuclear to cytoplasm ratio. So always remember the important characteristic of cancer cells or transformed cells is higher nucleus to cytoplasm ratio. Okay, so if we want to produce any product in any cell, first we have to check that whether it is having higher nucleus to cytoplasm ratio or not. If that is there, then that is transformed. And any product which is coming out of transformed cells is not safe usually, no? So that we have to check. And then fast growth. The growth will be very fast when compared to normal cells and cancerous cells or transformed cells. And they grow more in suspension. And they will have higher cell density. Okay. And they stop expressing tissue-specific genes. Okay. That means they are transformed. Now we have different types of cells and their characteristics, you can see, based on the morphology. That means shape and appearance or functional characteristics. There are three types of cells: epithelial-like, lymphoblast-like, and fibroblast-like. So epithelial-like means the cells which are attached to the substrate and they appear like flat plate-like structure or polygonal in shape, then they are epithelial-like cell types. Then lymphoblast-like means the cells do not get attached. They remain in suspension and spherical in shape. How the lymphoblasts or lymphocytes, how they look, round in shape, so like that they will be. Then fibroblast-like, the cells are attached to the substrate and they are elongated in shape and they are bipolar. You can easily distinguish the poles in that. So these are the different cell types. So here you can see in the epithelial how it looks like. Fibroblast is like bit elongated and lymphoblast is just round and they will be mostly in suspension or floating. So how can cell line be derived? So from any organism, whatever you want, like based on our interest, from any organism, we can isolate an organ. Then we have to culture that organ. Here it is, example is kidney. Then from kidney, that is organ. So we are taking a bit, not complete one. We are taking a part, that is tissue explant culture. Small tissue we are taking. So here it will have some tissue architecture. So the shape or properties architecture will be there. And or you can also cut the organ into very small pieces and we can culture them. That is primary culture. Or from this explant, tissue explant also, we can either do it manually or enzymatically. We can just separate the cells and we can split them, and we can, from the primary culture, we can split them and subculture into cell lines. So this is how we do cell lines. That is, we generate cell lines. So these are the human cell lines, how they look under microscope. So one stem cell line, one growing embryo. Here, scientists report a new method to make human embryonic stem cells that do not, as before, destroy the embryo. So that means here without destroying the embryo, we are separating the embryonic stem cells. So what is the mechanism? First is an egg fertilized by the sperm begins to divide. Okay, the fertilized egg or zygote. Then it will become 2, 4, 8. Okay, after the third division at the 8-cell stage, a single cell called a blastomere is removed. So here in the third step, you can see blastomere, only a single cell is removed from that particular ball of cells. And the blastomere is cultured with an established embryonic stem cell line. Okay, along with the stem cell line, they are culturing the blastomere and then separated to form new lines. Then here you can see left intact, the embryo continues to develop into a ball of some 150 cells, which will form a blastocyst. So the older stem cell technique removes the inner cell mass at this point, destroying the embryo. But here we are just removing a one cell and we are culturing that by co-culturing them with the stem cell. Okay, so this is how we can culture the embryonic stem cells without destroying the embryo. So here, what are the features? So what is the ploidy for continuous and finite cell lines? So for finite cell lines, they will be diploid, and continuous, they will be heteroploid. Okay. Then transformation is normal in finite, but they are transformed in continuous. So you have more number of DNA here in continuous. Then finite cell lines are anchorage dependent and density limitation of growth. That means like contact inhibition. Whenever the density increases, this inhibit their growth. But that is not seen in the continuous cell lines. They are anchorage independent as well as there is no density limitation of growth. And finite cell lines form monolayer cultures. They will grow in monolayers. Then continuous cell lines, they are monolayers or suspension. And the maintenance is cyclic for fine. That means we have to keep on checking, and that will be growing, then decline, growing, like that. It will be cyclic. But continuous, they will be steady state. There is no death. Okay. So they will be continuously growing. The serum requirement for finite cell lines, high requirement of serum. For continuous cell lines, there is low requirement. That means even without serum also, they will start growing. Okay. Then cloning efficiency. Cloning efficiency is low in finite cell lines, whereas it is very much high in transformed or continuous cell lines. Then the markers which we select, they are which they exhibit, they are tissue specific. Okay, because they will be they are not transformed. Okay, they will still exhibit the tissue specific markers of the source tissue from where they are isolated. Whereas the continuous cell lines, they exhibit chromosomal and enzymic markers. And virus susceptibility and differentiation may be retained in finite cell lines, but it is lost in the continuous cell lines. And growth rate is slow in finite cell lines, whereas it is rapid in continuous cell lines. Then yield is also low in finite and high in continuous cell lines. And the generation number, after some number of generations, it will stop. Then strain characteristics. So based on the strain, that will have some control features, how that mechanism will be controlled. So these are the different cell lines which are already present. Okay, by adding some growth factors, we can differentiate the embryonic stem cells into various differentiated cell types. So we have already discussed this yesterday. So animal cell lines and products. So what are the products which are already developed by using animal cell lines? So by using human tumor cells, angiogenic factor was developed. Then human leukocytes, interferon. Then mouse fibroblasts, also used for development of interferons. Then human kidney cell lines is used for urokinase production. Then duct embryo fluid is for vaccines for rabies and rubella. And human kidney cells are for human proteins. So what are the different applications of the cell lines? So they are used for screening of the anti-cancerous drugs. Then to perform cell-based bioassays, to determine the cytotoxicity, that means how a particular drug or a compound is affecting the cells, whether it is showing any toxic effect. Then in vitro screening of several drugs. Then production of antiviral vaccines. Then cancer research, where they are required the study of uncontrolled cell division in cultures. Then cell fusion techniques, to perform cell fusion techniques. The genetic manipulations, to study the effect of toxins and pollutants, and to study the function of nerve cells, chromosomal analysis. From the cells, this is amniocentesis. So that means from amniotic fluid, if we remove the cells and if we check the for chromosome analysis, we will know if there is any abnormality, okay, like that before birth itself. So these are the different applications of cell lines. So I will end this topic here, and now I will share another PPT where it is properties of tumor cells. Okay, so that is about the cell lines. So in cell lines, usually we have tumor cells or transformed cells or cancer cells or normal cells also. So now we will discuss about what are the characteristics of cancer cells. So this is about the cell cycle. So you all know the stages of the cell cycle: G1, S, G2 and M phase. So in the G1 phase, it is the cell growth phase. So this is all called as interphase till mitosis. G1, S and G2 together called as interphase. So G1, it is like a growth phase where it is preparing the cell for the replication of DNA. Okay, it is procuring all enzymes, proteins which are required for the DNA replication. Then here you have only n number of DNA, only one copy of DNA. Then after S phase, we will get 2 copies of DNA where the DNA replication takes place. Then in G2, again it is a cell growth phase, and which is preparation for the division or mitosis. So here the cell is same, which is having 2 copies of DNA. Now after mitosis, the number of copies of DNA, they will get separated to the poles. Then in cytokinesis, they will actually divide it into 2 cells. One cell is becoming 2 cells. So this is the cell cycle. Eukaryotes, in eukaryotic cell cycle. So what is a cancer? It is nothing but an uncontrolled division of abnormal cells in a part of the body which is called cancer. So that means this division is uncontrollable and the cells are abnormal. Only when they become abnormal, there is no checkpoint. That means in our body, every cell has it. They will receive 2 signals. One is to divide and second is to stop. So if even without receiving the signal for division, they starts dividing, then that can give rise to cancer. Or even after receiving the signal to stop the division, still they are dividing, even that can result in the cancer. So only 2 types of mechanisms. So here, because of loss of cell cycle control, that is the main thing. Because there are some checkpoints which are mediated, which are regulated by cyclin-dependent kinases. Okay, cyclins and cyclin-dependent kinases, enzymes. They will check that, okay, after DNA replication, are there any mutations? If they can be repaired, they will be repaired. If they cannot be repaired, then that cell won't continue to divide. They will undergo death. But if that phase is not there, then there is any loss of cell cycle control, then it can result in cancer. So before a cell divides, the DNA is checked to make sure it is replicated correctly. If it is not happening, a gene mutation occurs, then that can result in the cancer. So here we have another term we use is tumor. Okay, so tumor is nothing but a mass of tissue. It's a physical mass, abnormal mass resulting from uncontrolled division. So these tumors are two types, malignant tumor and benign tumor. Malignant tumors are cancerous. They will spread more. Okay, they are not restricted to any particular place. Whereas the benign tumors, they are non-cancerous and they are restricted to only local area so that they can be removed by surgeries. So first is malignant tumors. They invade or spread to the other parts of the body. They are very rapid and high rate of division can be observed. And from one place of origin, they can move to another places, which is called as metastasis. And cells travel through circulation and they reach another tissue and there they start growing. So if it enters into metastatic stage, then it is very difficult to treat because if one place it is stopped, it will start from the another place. Then what are the tumor markers? So how can we detect tumors? Means tumor cell markers are substances which are produced by the cancer.
What is mean by metastasis?
Metastasis is nothing but, suppose a person got some what you call lung cancer, suppose. So if they are treating the lung cancer, but again that person will develop liver cancer. So that means the lung cancer cells, they enter the blood and from blood they are traveling to the liver and there they are dividing, they are forming cancer. So that means spreading of cancer from not from the origin, but they are moving and going to another place and again they are developing to another type of tumor. So that stage is called as metastasis, which is very difficult to treat in that point of time. So the tumor cell markers, so the tumor cells will express some markers, okay, on the plasma membrane or in blood or in cerebrospinal fluid or urine. So there are some markers which you can detect only when there is tumor, okay, like hormones, then enzymes. So hormones only you can see during adrenal medullary because it is pituitary related hormones, no? So in adrenal medullary tumors, we can see that in blood, we can detect large quantities of hormones or like that, and some specific enzymes, some genes, some antigens, then antibodies. So like that we have some markers. So these are the hallmarks of cancer. So first thing is, like, how can you say that one cell is becoming, it became cancerous cell? So that means first thing is self-sufficiency in growth signals. That means though it doesn't get any growth signal, still it is self-sufficient and it will start dividing. Okay, that is self-sufficiency in growth signals. The second is insensitivity to anti-growth signals. That means though our body says stop dividing, but it become insensitive. So it won't listen. Okay. It won't react to the anti-growth signals, hence they will be continuously dividing. The third is evading apoptosis. So what is apoptosis? It is a programmed cell death. So all our body cells, whenever they are damaged or injured or in a normal turning over, like after some time, the cells will die. So that process is called as programmed cell death. So it is programmed. It is a normal process. Okay. Which will always keep a check to prevent the cancerous cells. But if that apoptosis control is lost, then it can become cancer. Then there will be no death for the cell and limitless replicative potential. So they will become like they'll be continuously replicating. Okay, so that is limitless replicative potential. Then sustained angiogenesis. So what is angiogenesis? It is the formation of blood vessels. So what it will do, like whenever it forms the tumor, it will secrete more and more of vascular endothelial growth factor, which is an angiogenic factor. Okay, so that will go, and there are some autocrine signaling. So that means, so in normally we have endocrine, paracrine, and autocrine. So here usually the factors are secreted from one type of cells, and that will go to another cell type where they will have receptors. But here, when that cell become cancerous, it will have the receptor and it will only secrete the factor. So that factor goes and binds to its own receptors and again secreting more and more amount of factor. So like that, it will secrete more and more amount of growth factors, and they will form blood vessels from the to connect with the body. Okay, and they will start getting all the nutrients from the body. So that means they are making them strong. The tumors are making them strong. So it is sustained angiogenesis, and then tissue invasion and metastasis. So it's the final stage where tissue is invaded. Okay, completely it is taken under control of tumors, and it metastasis, that means moving to another, it continuously taking over the body. Okay, from one place to another, it is spreading. So cancer metastasis. So metastasis is the spread of cancer to another location in the body. The new tumors are called metastatic tumors, while the original site is called the primary tumor. So it's the starting point where the cancer has come. So that is called as primary tumor. And after metastasis, after the movement to the another location, again the tumor which is developed, that is called as metastatic tumor. So here, usually to prevent the metastasis, the basal lamina of epithelium. So we have discussed now epithelium, how it looks. So it has a basal lamina, which is linked to connective tissue. So that basal lamina will stop these cells from entering into the tissue. But these malignant tumor cells, they break free of attachments with the adjoining cells, and every cell will be acting as a potent cancer cell to spread the cancer. And they go and attach to basal lamina, they secrete some enzymes which will digest the extracellular proteins, and they migrate into circulatory system. So like this, you can see above, like tumor cells are present in the epithelium, and we have the yellow one, which is basal lamina. So it is trying to stop, not to release the cancer cells, but they will secrete some enzymes which will break the basal lamina, and they will enter into the circulation. So through this circulation, they will go and enter into another, they will adhere to the capillary walls, and they will just squeeze into the capillary location, and then they will enter there. And again, they will form another type of cancer there. So here you can see like the same thing, which is involving some genes, all these were mentioned. So no need to go in depth. So that there are lots of markers. Okay, this is only explaining only of some type of marker, but you have lots and lots of markers. The next is benign tumor. So benign tumors, they do not spread to other body parts. Usually they are localized and they are small in size. And the growth rate is very slow. And they resemble and function the normal cells. Like we'll have normally warts. Okay, on our skin, we'll have small warts, which are benign tumors. If there is outgrowth means, then they are called as tumors. But they are not dangerous. They are very small, localized, and the growth rate is also small. Even some after many years also it will be of same size, right? And they usually look like normal cells only. They do not break out of originating organ, so they won't go anywhere, and they can be easily removed by surgery. So there is a classification of cancers. So cancers are classified based on the type of the cell from where they are originated. So they are called carcinoma, sarcoma, lymphoma and leukemia, germ cell tumor and blastoma. So what are carcinomas? They are the cancers which are derived from epithelial cells. So based on the origin from where they are coming, so the classification is given. So epithelial cells, if they are giving rise to cancers, they are called, you will get the name as a carcinoma after the end of the cancer. Okay. Hepatocellular carcinoma, like that. Example I'm telling. Then sarcoma. Sarcoma is the cancers arising from connective tissue, that is in bone, cartilage, blood, etc. Then lymphoma and leukemia, they are in hematopoietic cells. Then germ cell tumors, which are very dangerous usually, they are from pluripotent cells and they usually seen in ovaries and testicles. So they are called as seminoma in testicles if there is any tumor. Then in ovaries, dysgerminoma. Okay, and this can be actually like inherited from one generation to another, because that is germ cells. Then blastoma. So the cancers derived from the immature precursor cells or embryonic cells, they are called as blastoma. So what are the properties of cancer cells? So first thing is, there is uncontrolled mitotic divisions causing unorganized growth. The normal growth is very organized. So we have a specific shape, specific size, structure, like that. But in cancer cells, it is uncontrolled and it is unorganized. Then next is, due to uncontrolled growth and division of cells, a tumor is also called as neoplasm. Okay, and they are far less adhesive than the normal cells. They usually try to move from one place to another. And they exhibit a number of alterations on cell surface in the cytoplasm and in the genes. So that is nothing but mutations or transformations, even in the cell surface, cytoplasm and the genetic composition also. They do not undergo differentiation. So once they become cancerous cell, there is no further differentiation. Then they lose the ability to communicate with other cells through chemical signals. Okay, there is a signaling, cancer cell signaling. And they also lose sensitivity to anti-growth signals from surrounding cells. That means they will stop, like if other cells, that is called loss of contact inhibition. So usually in the contact inhibition, if there is any outgrowth, the adjacent cells will be telling to stop. Okay, that is, they will give the signals. But here the cancer cells will lose the capacity to stop even after getting the signals from the surrounding cells. And they lose the adhesion molecules that keep them bound to neighboring cells. So when they are removed, okay, the adhesion molecules are removed, then there is no more attachment and every cell can freely act. And the cancer arises from the loss of normal growth control. So if the normal control is lost, then simply it can give rise to cancer. And cancer can be a genetic disease, or it can be spontaneously generated also based on the lifestyle or exposure, like that. So you have inherited cancers or sporadic cancers. And cancers typically involves a change in gene expression or function. So they are called as qualitative change, quantitative change. So how much amount, if we are looking, if there is any change in growth of like there is any enlargement of some organ or like that, then that is quantitative. The number is increasing. Whereas qualitative means if more expression of proteins or if there is any change in the phenotype, so that is qualitative change. The lack of contact inhibition, the contact inhibition will be completely lost. And loss of limitations on the number of cell divisions. So usually the normal cells will have a limitation for cell divisions, but cancer cells will lose that limitation. And it has the ability to grow in culture. Okay, so normal cells, they do not grow well in culture, but cancer cells grow much better than the normal cells. Then in laboratory cultures, normal cells divide only when attached to the surface, but in these cancer cells, they can also circulate in suspension. They can grow in suspension. Then angiogenesis. So it is they secrete substances that cause blood vessels to grow towards the tumor. So this is how they look, normal cells and the cancerous cells. So normal cells, cytoplasm content is different. In normal cells, it is more. In cancer cells, it is less. Then nucleus is more, nucleus is less in normal cell, small size, but it is more amount of nuclei and more number of nuclei also you can see in the cancer cell. Then nucleolus also differs, chromatin content also differs. So this is how the difference is. So this is how it looks like. Regulated cell growth, they are very much organized. Whereas in cancer cells, you can see unorganized and outgrowth is seen. And this is how, like loss of normal growth control. So how it is, like normal cell division, how it looks like after some generation, if there is any cell damage, then it will usually suggest for repair. If repair is not possible, then the cells will undergo suicide or apoptosis. But in cancer cells, the mutations get accumulated within the cells. Okay, so finally,
excuse me, ma'am. Yeah. One cell may contain 2-3 nucleus. That will happen.
Yeah, that happens.
In cancerous.
Yes. Okay. So it will become multinucleated.
Okay.
Okay, so that means very rapid. See, it won't be always there, like it won't show 2-3 nucleus, but it will be rapidly dividing. So when you see at every point of time, when it is dividing, so there will be always continuously, you can see 2 or more nuclei and they will get separated. Again, in each cell, you will see 2 or 3 nuclei, again they will be separated like that. So its uncontrolled growth can be seen. So here another type of representation. So here you can see the cells anchor to dish surface and divide. So that is called Anchorage Dependent. So this is the confluency, what I was telling. Like in the first dish, you can see only 2, 3, how many? 1, 2, 3, 4, 5 cells. But in the next dish, you can see they formed a single layer. Okay, so that means it is the 100% confluence. Okay, just I wanted to explain that term using this image. So when come to normal cell versus cancer cells, so the cells anchor to the dish surface and divide. That is anchorage dependent. And when cells have formed a complete single layer, they stop dividing. This is called as contact inhibition or density dependent inhibition. And if we remove some cells for subculturing or something, or by scrapping, you will get some space and again they will fill the new cells. So if some cells are scrapped away, the remaining cells divide to fill the dish with a single layer and then stop. This is called density dependent inhibition. And here even the pH also will change. And cells in culture and in vivo exhibit contact inhibition, whereas cancer cells, they won't have any feedback mechanisms or they will form clumps and foci. So what are the causes of the cancer? Okay, so till now we have discussed about the characteristics of the cancer cells. So now we'll discuss what are the causes. So the great majority of cancers, that is 90 to 95% of the cases, are due to environmental factors like smoking or alcohol or exposure to some rays, UV rays, X-rays. So like that, their lifestyle, unhealthy habits. So and also more exposure to carcinogens. So all these can result in the cancer, 90 to 95% of the cancers. But 5 to 10% are inherited. They will be inherited or genetical. Okay, that is only 5 to 10%. So what are the environmental factors? So in this, 25 to 30% are contributed by tobacco, and 30 to 35% are through diet and obesity. Then 15 to 20% for infections, through infections, some viral infections are like that. 10% are through radiation, both ionizing and non-ionizing radiation. Then pollutants, stress. So all these also very minute quantities, they can get involved in propagation of cancers. Then inherited genetics. So cancer is fundamentally a disease of tissue growth regulation failure. That means the regulation of growth regulation is failed in a tissue. Then that can result in the cancer. So in order for a normal cell to transform into cancer cell, the genes that regulate the cell growth and differentiation must be altered, right? So the affected genes are divided into two broad categories. They are oncogenes and tumor suppressor genes. So what are oncogenes? Oncogenes are nothing but they will be usually inactive. Oncogenes are present in our body and tumor suppressor genes are also present in our body. But oncogenes are those genes which will be inactive in our body. They are proto-oncogenes in the form of proto-oncogenes. And tumor suppressor genes, they will be always active in the body so that they will suppress, they will always keep on watching if there are any tumor cells. And if there are any tumor cells by mistake, they will counter them. But if there is activation of proto-oncogenes, or if there is any suppression in tumor suppressor genes, then it can result in cancer. So oncogenes are genes that promote cell growth and reproduction. And tumor suppressor genes are genes that inhibit cell division and survivals. So if there is any mutation in the tumor suppressor gene, then what happens? The checkpoint for increased synthesis from entering G1 to S phase, there will be inhibition that will be lost and continuously that will be dividing. Okay. And when this oncogene, proto-oncogene gets activated into oncogene, then also there will be increased risk of cancer phenotype because that will induce the cells to synthesize more and more amount of cells. So here the analogy here is, here you can see the normal genes are there, 2 copies, and there is one mutation. If there is only one mutation, they can be carriers, but necessarily they might develop the cancer or they may not. But again, second mutation comes, then it can result in cancer. So here you can see this is a CAR, which is tumor suppressor gene. So this another one is activate oncogene. So when this tumor suppressor genes are lost and active oncogenes get activated, then the brakes are lost. When there is lost in brakes, then there will be accident here. Okay, so that means development of cancer. So what are the signs and symptoms? So when cancer begins, it produces no symptoms initially. Please mute yourself. Somebody's mic is. Yeah. So when cancer begins initially, there won't be any symptoms or signs. Only after the tumor starts growing more and more, then they will develop some symptoms and signs. So here, there are 7 warning signals. Okay. One is change in bowel or bladder habits. We have to keep a check on bowel movements and bladder habits. Then a sore that does not heal. If there is any sore or something, if it is not healing usually, and unusual bleeding or any discharge. Then thickening or lump formation in the breast or anywhere, any part of our body. Then indigestion or difficulty in swallowing. Then obvious change in the wart or mole. That means usually warts will be very small and you cannot see that much growth. Every day you don't see a wart growing or a mole also. But if it is cancerous, it will be spreading or it will be increasing in its size. If there is some change in that, we have to immediately go to the doctor. Then nagging, coughing, hoarseness. So like that, these are some warning signals of the cancer. Then cell deaths. There are 2 types of cell deaths. One is apoptosis and other is necrosis. So apoptosis is nothing but the programmed cell death. So it's a tightly regulated form of cell death, also called as programmed cell death. It's a normal process. Morphologically, it is characterized by chromatin condensation and cell shrinkage in early stage. So what happens whenever the cell, there is a signal for a cell to undergo apoptosis, there will be chromatin condensation, followed by cell will start shrinking. Okay, and then the nucleus and cytoplasm will fragment, and membrane-bound apoptotic bodies will be formed. And if that morphological changes occurs in that cell, then another type of cells called phagocytes, they will come and they will eat up. Phagocytes means cell eating. They will eat these cells. So this is initiated by signal transduction process. There will be a fast ligand, this death ligand, which will appear at the top of the cell, which has to undergo the apoptosis. Therefore, the cells which have the receptors for this fast ligand, that will come and bind to this apoptotic cell, and then they will engulf. And then there will be no cause of inflammation. Okay. And ends with the fragmentation of cells into smaller bodies. So that means similarly, like whenever I will just give you the analogy, like example. There are deaths like suicidal death or normal death and accidental death. So in suicidal death or normal death, it won't affect anyone. Okay, only that person will die. So in the same way, when apoptosis is there, only that cell will die without affecting anything. Okay. But in necrosis, it is like an accidental death. So when a person do the accident, it may cause or it may injure some other people also. It can cause it will definitely cause some damage, whether it is like financial or life or something like that. Okay. So here also, whenever the cell undergoes necrosis, that will cause a lethal injury. That means cells undergo another form of cell death called necrosis, where the cells will swell and they will rupture, and it will release all the intracellular contents into the surrounding cells. Therefore, it will cause inflammation in the surrounding cells. So it's not not a programmed cell death. It is unprogrammed cell death. So it can happen only during some infections. So it is initiated by direct cell damage, mostly physical. Okay, if there is any injury, what happens at that stage, at that particular place, there will be necrosis, cause inflammation, and ends with the total cell lysis. Not only that cell, but all surrounding cells also will get affected. So these are the features of necrosis and apoptosis. So in the necrosis, the cell size will be enlarged, they will swell. While in apoptosis, it will shrink, the size will be reduced. Then nucleus. So in apoptosis, it will shrink. The nucleosome will become fragmented. Whereas here also it will become fragmented, but it will be lysis and that will be released outside. Then plasma membrane also, it is in necrosis, it will disrupt. That means it will burst. Whereas in apoptosis, it will be intact. And the cellular contents in the necrosis, they will be leaked out and enzymatic digestion happen. And in apoptosis, they will be intact and released into apoptotic bodies. So is there any adjacent inflammation in the necrosis? Yes. But in apoptosis, we don't see any adjacent inflammation. And physiological or pathological role, necrosis is usually pathological. That means any damage occurs. But apoptosis can be both physiological as well as pathological. And other important points of apoptosis are their controlled death of individual cells. They're induced by physiological stimuli, no inflammation, shrinkage of cytoplasm and condensation of nucleus. Then blebbing of plasma membrane. The plasma membrane will form a blebs, small blebs. There is no loss of integrity and ATP is required. So it's an active process. Energy is required. And we'll have functional mitochondria because ATP is generated from the mitochondria. Therefore, mitochondria is involved. And cell death pathway activation happens in apoptosis. Whereas in necrosis, it affects a group of cells evoked by non-physiological events like viruses or toxins, etc. Then inflammation. It can cause inflammation of the surrounding cells. Then swelling of the cytoplasm and mitochondria. Loss of plasma membrane integrity, no energy requirement. It's a passive process. And calcium overload is the key feature. If there is more amount of calcium, then it can be necrosis. Then types of cancer. So at the organ levels. First is lung cancer. So in 2013, 1,74,470 people died of lung cancer. And women also they are died of lung cancers and breast cancers. So the symptoms are persistent cough. There will be persistent cough. Then blood may appear in sputum and chest pain. The treatment can be surgery if it is localized or radiation therapy and chemotherapy. And most common causes are smoking and tobacco chewing. So if we avoid smoking and environmental tobacco smoke, we can prevent the lung cancers. The next is breast cancer. So 1 out of 8 women, okay, they develop breast cancer. And as the age increases, then there is a lot of chances of getting the breast cancer. So usually we can detect the breast cancer by self-checking of breasts, as well as we can go for mammograms. After some age, regular checkup is required. And the symptoms are lump in the breast, thickening or dimpling of skin, dimpling of breast, then skin irritation, and distortion or tenderness. If there is any change in the color or sensation or anything like that, it is always better to consult a doctor. So the risk factors are family history, hyperplasia, then long menstrual history, then obesity after menopause, and oral contraceptives. The treatment can be removed, okay, lumpectomy or radial mastectomy, radiation, chemotherapy, etc. Prevention is exercise. So if we lose the weight, then automatically the risk factor will be decreased. Then colon and rectal cancers. It's the third most common cancer in women and men. And the risk factors are, if obese or above 50 years of age. Then family history of colon or rectum cancers or polyps. Then diet, which includes high fats or low fiber. Then smoking, high alcohol consumption, lack of exercise. So all this. So 90% can be preventable. Treatment includes radiation, surgery, and chemotherapy, and regular exercise, a diet heavy in fruits and plant-based origins, then healthy weight, moderate alcohol consumption. So all these are, they can prevent from the colon and rectal cancer. Then prostate cancer. So it's the most common cancer in American men, like excluding the skin cancer. So here it is muscular and walnut-sized gland that surrounds the part of urethra. So its primary function is to produce seminal fluid. So the symptoms here are, we don't find any specific symptoms. They are non-specific symptoms. Weak or uninterrupted urine flow, difficulty in starting or stopping urination. And the risk factors include age, race, nationality, family history, diet, lifestyle, and vasectomy. And prevention can be by high inclusion of high lycopenes in diet and vitamin E. Then skin cancer. So long-term effect to the sun. Okay, that can result in skin cancer. So there are many types. So out of that, malignant melanoma is the deadliest form of skin cancer. So sun usually gives 3 types of harmful rays: UVA, UVB, UVC. We get exposed more and more, then that can result in cancers. The prevention is limited exposure to harmful UV rays, drink more fluids than usual, apply cool compresses to skin, or moisturize skins or sunscreen lotions, so like that. And what to look, that is ABCD rule. A means asymmetry. Half of the mole does not look like the other half. The symmetry will be different for moles. And then border irregularity. So you can see here in the image, the border is uneven in second image. Color. Pigmentation is not uniform. It is not of single color. You can see different colors. Then diameter. So if it is greater than 6 millimeters of diameter, then we have to go for testing. Then testicular cancer. So it affects usually 8,250 young men in 2013. So it will change, the statistics will change year to year. And men between the ages 15 to 35 are at greater risk. And so the important thing is to practice regular testicular self-exams. And Lance Armstrong Foundation, that is Live Strong, it campaigns regarding the awareness of the testicular cancer. Then ovarian cancer. So it's the 5th leading cause of cancer deaths among women. And the most common symptom is enlargement of the abdomen. The risk factors include family history, age, childbearing, or cancer history, then fertility drugs, then talc use in genital area, and genetic predisposition. So diet, which is rich in vegetables and low fat, exercise, sleep, stress management, and weight control. So all these can prevent from the cancer. Then cervical and endometrial or uterine cancer. So here the test is pap smear test. So cells from the cervical region are isolated as a smear and they will be checked. The risk factors are for cervical cancer, early age at first intercourse, and multiple sex partners, then cigarette smoking and sexually transmitted infections can cause cervical cancer. Then endometrial cancer includes, the risk factors includes age, endometrial hyperplasia, overweight, diabetes, high blood pressure. So all these are endometrial cancers. risk factors. So like this, you can see in the normal one, stage 1B, late stage, it will keep on spreading. Then other cancers, they include pancreatic cancer, which is silent, and only 4% of survival rate can be seen. That is only 5 years. Then leukemia, it is a cancer of blood forming tissues. It also spreads very fast. So this is all about the cancer cells. So we will continue the other topics in the next class. So if you have any doubt, sporadic means spontaneously, they will simply come out. Okay, it is not like inherited or like that. Yes, any doubts?
No, ma'am.
Everything is clear. You can, if you have any doubts from the previous classes also, you can ask.
Ma'am, you please send these slides in the group, ma'am.
Yeah, I am not there in the group. So if you can add me, or at the last class, if you can just remind me at that point of time, I will send.
Okay, ma'am.
Okay, ma'am. Okay, just one class before the last class, you tell me. Okay, so that I will prepare all these things and I will send.
Okay, ma'am.
Okay.
Okay, ma'am. Thank you, ma'am.
Yeah, if there are no questions, I'm stopping. Okay, we will continue in the next class.
Okay, thank you, ma'am. Thank you, ma'am.
Thank you, ma'am. Thank you.
Thank you.
Thank you, ma'am.